नमस्कार दोस्तों, आपका स्वागत है आपके ही यूट्यूब चैनल फार्मेसी वाव में। आज जो टॉपिक डिस्कस करेंगे उसका नाम है स्टीम स्टलाइजर। डेफिनेशन: स्टलाइजर द एक्ट एट प्रोसेस फिजिकल और केमिकल दैट डिस्ट्रॉय और एलिमिनेट ऑल वाइबल माइक्रोब इंक्लूड रेजिस्टेंस बैक्टीरिया स्पोर फ्रॉम fluid or a solid. For example, sterilization method are steam treatment at 121 degrees centigrade or dry heat at 230 degrees centigrade, flushing with sterilization solutions such as hydrogen peroxide or ozone or radiation and filtration. Sterility: sterility the reduction of antiseptic level of contamination in the load to the point where probability of sterility is less than 10 to the minus 6. D value: the time in minute required for a log reduction or 90% reduction of a specific microbe population under the specific lethal condition. For steam sterilization is determined at constant temperature. Z value: the number of degree of temperature change necessary to change the D value by a factor of 10. The F value is a measurement of sterilization effectiveness. F is defined as the equivalent time at temperature T delivered to a container or unit of the product for the purpose of sterilization, calculating using the specific value of Z. F0 value: the term F0 is defined as the temperature of equivalent minute of the steam sterilizer at temperature 121 degrees centigrade delivered to a container or unit to product calculated by using a Z value at 10 degrees centigrade. Method: vacuum cycle, a more effective method by means of mechanical vacuum pump or steam educator. Vacuum as a low 15 to 20 mm of Hg. Apply for 8 to 10 minutes. दोस्तों, स्टीम स्टलाइजर में डिफरेंट तरीके से अप्रोच रहता है साइकिल डिजाइन करने के लिए, जस्ट लाइक ए प्री वैक्यूम साइकिल. Pulsing cycle, a series of alternating steam pulse allow for vacuum excretion. And third one is a air steam mixture. Terminal sterilizer for large volume parental. All injection required to compensate the great expansion of air or nitrogen in the heat into the head space over the fluid. Well mixing chamber, fan, raining effect or external pump for cooling. Cycle developed. considering factor into account. Nature of the load, porous material, heat sensitive material of the product, type of the installation, employed container closure, heat stability product, overkill approach, heat legible product, bioburden approach, bioburden studies, number of microorganisms, D value studies on highly resistant spore form, included the spore into the actual solution for solid material and steps. Preparation for validation, temperature sensor device, T-type thermocouple, ensure the case of flexibility sheet, platinum grade of wire having 0.1 degree centigrade accuracy, temperature standard, RTD traceability of the National Bureau of Standard, calibration of thermocouple, to temperature 0 degree or 130 degree centigrade, corrective action, stability, accuracy. Stability is a 0.03 degree centigrade and accuracy is a 0.5 degree centigrade. Preparation for validation. ऑटोक्लेव वैलिडेशन के लिए ऑटोक्लेव रेडी होना चाहिए, वैलिडेशन नोजल एंड एडेप्टर अवेलेबल होना चाहिए, डाटा लॉगर होना चाहिए, डाटा लॉगर आउटपुट एंड मल्टीपल चैनल डिवाइस होना चाहिए, BIs होना चाहिए और लोड होना चाहिए। वैलिडेशन प्रोटोकॉल रेडी होना चाहिए, वैलिडेशन प्रोटोकॉल इंक्लूडेड ऑब्जेक्ट ऑफ वैलिडेशन, रिस्पॉन्सिबिलिटी ऑफ वैलिडेशन, personnel and operation department personnel, identification of description of sterilizer and its process control, identification of SOP equipment, calibration of instruction, SOP and description, identification and calibration of the temperature monitoring equipment. Validation protocol included BIs of studies, empty chamber heat distillation studies, container mapping studies, load container heat penetration studies, microbiological challenges studies, evaluation of drug product, cooling water if applicable, integrity of vent filter. हीट डिस्ट्रीब्यूशन स्टडी क्या होती है? हीट डिस्ट्रीब्यूशन studies to demonstrate the temperature instability of the stability of the sterilizer medium through the sterilizer. Conduct on both empty and loaded chamber with maximum and minimum load configuration. इसके लिए सबटेक क्राइटेरिया क्या होता है? लेस देन +1 डिग्री of the main temperature. Conduct 3 run to obtain constant result. Distributorship of the thermocouple, exit drain adjustment of the control sensor at last 10 pore, normally 15 to 20 probe. Heat distribution study क्या होती है? A load chamber heat distribution test. The thermocouple should be positioned in the same location used for empty chamber heat distribution. Avoid containing solid surface. Do not place within any container. Data should be obtained in regular interval. Container mapping क्या होता है? To determine the coolest point within the liquid filled container. Temperature mapping should be conducted on the different container type, size and fill volume to be validated. The number of the thermocouple used depend on the container volume. Possible to use a single thermocouple at different position and can be conducted in the similar autoclave or report. Heat penetration study क्या होती है? To determine the coolest point within the specific load and configure and to assure that these points be considered exposed to sufficient heat lethality. Prior to conduct heat penetration studies, determine maximum and minimum load configuration. Probe container at cooled point should be disturbed. Probe container at cold spot should be distributed uniformly throughout the load. Penetration thermocouple are positioned at point within the process equipment specified to be most difficult to steam heat penetration. Microbial challenge test study. Microbial challenge study में क्या करते हैं? Biological challenges are employed during the heat penetration study in order to determine the degree of process lethality provided by station cycle. Microorganism frequency utilized, overkill, Bacillus streptothermomellus and बायोब्रेन के लिए हम कैलिबेटा BI यूज करते हैं इंस्टेड ऑफ ई कोलाई. टाइप ऑफ BI, spore strip or spore suspension in the suspension medium. Molecular challenges study are conducted concurrently with heat penetration studies. Validation report. Common element of the all report, identification of task report by number, reference to protocol, a brief summary of range of operation condition, experiment and how they are controlled, a procedure for maintaining control within the approval range, a summary and analysis of the experimental result, a brief description of any deviation. Conclusion. Review of the approval cycle development report are not usually for the part of validation report.